I welcome everybody who is interested in science, and today I will discuss a paper where researchers exhibited how a targeted simulation of a human brain with direct current can boost creative thinking. If you want to know more about this paper and first publish practical applications on humans, stay tuned and you will know all the details. Let's go!
What are you waiting for? Chinese New Year? Go!
The formation of rules, or scientifically speaking cognitive templates, help you to solve faster tasks of the same type. However, in the new environment, such stereotype can significantly reduce your productivity. Previous studies showed that the learning of rules may be related to the functioning of the dorsolateral prefrontal cortex, shortly named DLPFC. For instance, patients with focal lesions in this area of the brain exhibited more creativity in matchstick puzzles, where non-template original thinking was required. Thus, DLPFC is supposed to be responsible for applying the learned cognitive rules. Then researchers decided, let's alter the activity of DLPFC and check whether these manipulations will affect the performance of the participants during cognitive tasks. In order to increase or decrease the activity of DLPFC, scientists used Transcranial direct current stimulation technique, shortly named TDCS. TDCS is a form of neuromodulation that uses low direct current delivered via electrodes placed on the head of a participant. Depending on the direction of the applied current, TDCS either facilitates the propagation of electrical signals in the brain, a nodal configuration, or hinders the signal transmission. Such mode is named cathodal.
I get ya!
Now let's refer to the actual experiments. Participants were asked to solve matchstick arithmetic problems of several types that differ depending on the requirements of breaking previously learned cognitive rules. More a puzzle relies on breaking previously learned rules, the harder the puzzle is to solve. Let's take a look on some examples to make it clear. Type A. These puzzles are solved by moving one matchstick within a specific numeral. In this case, numeral 4 turns into 6. This is the easiest type as the solver has to override only value constraints, which are essentially loose chunks. Thus, in this case, only one loose mental constraint should be broken. Type B. This type is solved by moving one stick from the plus sign to the numeral. Its difficulty is intermediate as the solver has to override both value and operator constraints, and it requires decomposition of the plus sign and the values. Therefore, two previously learned rules should be overridden.
Actually, it wasn't that hard.
Finally, type C. This type is solved by rotating the vertical stick of a plus sign, changing it into an equal sign, thus creating a tautology. This is the hardest type to solve as an individual has to override both the operator and tautology constraints, and it requires decomposition of both the plus sign and the equal signs. Thus, the participants has to rethink the whole framework of the problem and then break a sequence of previously formed mental constraints, all within one movement of a matchstick. In many cases, participants tried to use a solution hypothesis similar to those from type A and B and therefore could not solve the problems. Here, you really have to find an original idea.
Find a truly original idea.
The actual experiments consisted of three parts. Firstly, participants were asked to solve 12 puzzles. 2 minutes were assigned for each problem. Then the brain stimulation was applied for 15 minutes. Recall, either cathodal or nodal stimulation, corroborating or hampering the activation of DLPFC respectively, or sham, which is a control condition when the applied current is zero but all electrodes are still mounted and connected to the stimulator. After the brain stimulation, participants were asked to solve other 12 puzzles, but of the same type as before, plus the puzzles they could not solve in the pre-stimulation session. Now let's refer to the results of the post-stimulation phase. Has the applied simulation changed something? For the problems of type A and B, the easiest and the medium ones, the application of TDCS did not cause any differences. Has the experiment miserably failed and the researchers just wasted the money of taxpayers?
You're fired.
Not so fast. Let's go to the results of type C, the hardest puzzles that require the strongest break of cognitive constraints, and to turn inequality to a tautology, basically to break the framework of a puzzle. Let's see the percentage of people who solve the task after the application of each type of the stimulation. If our brain stimulation doesn't cause any differences, the values should be the same for all types, cathodal, nodal, and control, scientifically named sham. What are the results? After sham, about 5% of the participants solve the problem. The same is for a nodal stimulation activating DLPFC. But recall, our hypothesis is that the effect should come after the inhibition of DLPFC. Now, a key moment: will the percentage of the people who solve the task increase after we suppress the activity of the brain region mediating rule application? Yes, it did. 32% of participants were able to solve the task compared to 5% for 2 other conditions.
Yes!
These results revealed that suppressing the activity of DLPFC by cathodal TDCS has indeed helped to break the cognitive rules or templates, thus increasing the creative thinkings and the chances to solve the third type of the puzzle. Importantly, these results were specific for the hardest type of the puzzle, and the applied stimulation didn't affect the performance for the first and the second groups of the puzzles. This additionally supports the idea this inhibition of DLPFC is beneficial to break previously learned mental constraints, but for the task where this is not critical, this break is not a decisive criterion. So it's not necessarily bad to apply some rules, but sometimes you need to get rid of them.
Sort of exciting, isn't it? Breaking the rules.
So if you like this video, please give it a thumb up and do not forget to subscribe to the channel, allowing all notifications not to miss new and cool scientific stuff. Also, if you have any ideas and questions or just want to share your emotions about what you've just seen, feel free to post them in the comment section below. Every comment will receive a response. This is for today, and I hope to see you soon.